Hi everyone, welcome to the March 2022 Twist Bioscience webinar, Expanding the Toolbox for Agrigenomics. My name is Paul Doran, the Director of Business Development for Twist Biosciences. I recently joined Twist through the acquisition of iGenomics, which is a startup from the Scripps Research Institute focused on developing next-generation sequencing tools such as high-throughput library preps, which we're going to talk about here in a second. Before we start, I'm going to go over a few housekeeping items. First is all lines will be muted during the webinar. We will answer all questions after the presentation. So if you have a question, please submit it to the Q&A box rather than the chat box. Following the presentation, if you have a minute to take a brief survey, we'd really appreciate your feedback. So the agenda, I'm going to do a short intro to Twist. Do an overview of the new 96-plex, which previously was the Riptide assay from iGenomics. Then talk about some low-pass sequencing applications with 96-plex, and then introduce our main speaker, Dr. Josh Clevenger, who's going to talk about his KUFU pipeline. So what's unique about Twist? It's our DNA writing platform. Our technology enables us to make an enormous amount of high-quality DNA at scale. So if you look at what most people are doing, they're doing things on a 96-well plate and they can make one gene. If you look at Twist's ability to print millions of oligos in a single run enables us to make about 9,600 genes at a time, which is about 10,000x more scale and throughput. So with that technology and that capability, what can you do? So we are producing genes, we're making oligo pools, we're making variant libraries for screening, and then relevant to this presentation, oligos are the basis for pretty much every NGS product. And then next is biopharma, where we're making vaccines involved in antibody development. And then last, we're leveraging our ability to write DNA for data storage, which has sparked collaborations with companies like Netflix and Microsoft. So in the NGS space, we are broadening our offering and expanding what applications we're addressing. Currently, we've got human exome, we've got fixed panels, we've got custom panels, library prep reagents and kits, synthetic assay controls. I think a lot of people are familiar with our COVID controls, targeted methylation, that's enabling much lower input than previous methods. And then we're going to focus primarily on low pass today, and there may be some additional webinars in the future here where we'll start talking about population genetics and plasmid QC that will be enabled by the 96-plex. Okay, so an overview of 96-plex library prep. So what's fundamentally different about this library prep? There's at least a few things, and one of the main things that people like about the technology is there's no fragmentation and repair or ligation. It's also tunable for high and low GC content. You can automate the assay, but in a lot of cases it's not necessary. So little to no automation is needed. And you can do anywhere from 96 samples at a time up to 960 samples that can all be run on a single flow cell. So how does it work? So you just take your DNA template, you put it into a 96-well plate. The first set of reactions, the A reaction, are run individually. So you have a random primer here, followed by a sample barcode, followed by a partial P5 lumen adapter. This hybridizes, and then it's an extension reaction, which is then terminated by dideoxys that are biotinylated. So that enables you, with a biotin on the end, to do a capture. Before you do the capture, you pool everything into a single tube. After the capture, you do a wash, and then now you're ready to accept the second adapter. That's added using a random primer. This is the partial P7. It extends out, and then the final step is to do a few rounds of PCR with the full adapter primers. The primer on the P7 side actually has a plate barcode that allows you to put multiple plates into a single sequencing run. There is a size selection step, so depending on if you're going to do 2 by 150 or 2 by 250, you can select what size you want to target, and you're ready to go. So it's very simple high throughput assay, and it's enabling things like low pass, and that's what I'm going to talk about next. So the first thing is doing, and it's a couple examples where you're doing mapping recombination breakpoints, and this is going to be in sorghum. So looking at LD decay in plants, it's been determined that more markers are needed. So if you look at GBS and microarrays, we're only capturing about 30 to 40% of the potentially detectable loci. So to saturate the genome of sorghum, you need about 700,000 markers, and with maize you need somewhere in the neighborhood of about 2 million. So back at PAG in 2020, which was probably the last time many of us actually met at a conference on agrogenomics, Dr. Schnabel presented on using more markers and what it could mean. So this is a GBS experiment that was done back in 2017. They weren't able to find the region associated with starch content. They reanalyzed the data using 2 times more markers, and were able to find the region in chromosome 2. It basically bumped up the magnitude by about 2 orders. In another example that he presented at PAG was a paper that was from 2018. They replicated this using low pass. And this is just a comparison of the number of SNPs that they got between the two. The number of markers was about 14 times what they got with GBS. And then after they did filtering, it was about 19 times more really high confidence SNPs. And what that translates into is identifying 7 times more segregating sites using this method. The next one I really think is fascinating in terms of what you can do with it. So I worked at Affymetrix for about 8 years and did a lot of things with SNPs. So really was involved in quite a few GWAS projects. This particular project was done with Matt Hunselman at TGen. They had done a canine GWAS in using microarrays and didn't find anything. So we tried to do just a small pilot and ended up finding some things that were really interesting. So we only ran 1 plate, and with that plate we were able to pull up, using low pass, 5 million variants, which is about 50 times more than the leading canine array. We were able to improve the association signals to the point where there was a region that was worth investigating, and underneath the peak they were able to find candidate loci that really match well with the biology of idiopathic pulmonary fibrosis in humans. So what's really cool is after they identified the peak, they just said, well, we've already done the sequencing. Let's see if we can find new variants using the sequencing data. And that's what they did. They were able to find new variants only in the homozygous risk haplotype carriers. And so this basically allows you in one prep to do GWAS, fine mapping, and variant discovery. Okay, the last one I want to mention, and this one I think even takes it a step further because it adds structural variation, and you don't necessarily need a reference, and that is KMER mapping, using KMERs to do mapping, discovery, and diversity analysis. So there's a paper in 2018 from Raman that came out, and a lot of folks have used that to try this method. Basically what you do is you do your sequencing of case controls, you count KMER frequencies, you find KMERs that are associated with the cases and controls, and then you can assemble those KMERs and then map them back to the genome, or if you don't have a genome, you will have something to map once you have a reference. So more recently, a couple years later, you got Wojciech came out in 2020 where they looked at Arabidopsis using this to try to see how does KMERs match up with SNPs. And what they found is there's very high correlation between the two, where they got 458 phenotypes were identified using the two different methods, but there was 129 that were unique to KMER and 137 that were unique to SNP. Now, if you look at the significance of KMERs versus SNPs, KMERs tend to be more significant when you compare the two. The nice thing about it is that because you're using low pass, you can identify SNPs using low pass and then go back and do KMERs. So with one experiment, you can actually get all of this. So here are the applications with the KMERs. You can do GWAS, you can do diversity analysis or population stratification, which essentially is just using KMERs and principal component analysis. It works with and without a reference. Utilizes unmapped reads, which is likely where if you have something that's unmapped and it's got a unique trait, there's probably a good chance that it's within the unmapped reads. It detects major deletions, insertions, and rearrangements. And like I said, you can do SNP-based analysis, which then enables you to do fine mapping and SNP discovery. It is my privilege to introduce Dr. Josh Clevenger. We've worked closely over the last year and a half. He was trained in the lab of Peggy Osias Akins in plant breeding and genomics at the University of Georgia's Institute of Plant Breeding, Genetics, and Genomics. During his PhD, he was a major contributor in the Peanut Genome Initiative. After a short USDA NIFA postdoctoral fellowship, Dr. Clevenger worked for Mars Wrigley on a global nut science team where he designed and deployed genetic improvement across 5 continents. Dr. Clevenger joined Hudson Alpha in the spring of 2020 as the faculty investigator in the Center of Plant Science Sustainable Agriculture. Specifically, Dr. Clevenger's lab is focused on optimizing the computational pipelines for the use of next-gen sequencing and genomics in the genetic improvement of crops with complex genomes. So without further ado, here is Josh.
Hi, I'm Josh Clevenger, faculty investigator for Hudson Alpha Institute for Biotechnology. I'm really excited to talk to you about how low coverage whole genome sequencing really is changing the way that we think about genomics assisted crop improvement, especially in minor crops. And so the structure of the presentation is really, I want to talk to you about my journey searching for genes to improve peanut. So, and this is applicable across many minor crops. And then how we came across Riptide Library Prep and how we're using it and how it's really changing the game for us and for our collaborators and partners. And then I want to speak a little bit about how I see the future of utilizing whole genome sequencing in breeding applications. So in terms of lessons from peanut, peanut is a complex and simple genetic crop. It is a segmental allotetraploid, which means that it does have 2 distinct genomes, an A and a B genome, that segregate independently more or less when it is fertilizing. But there are regions of those chromosomes that are so similar that they actually interact, turn meiosis. And so they can have some crossing over between what we call homologous copies. So the A copy and the B copy that are similar to one another. And so that causes these sort of pockets of segmental or auto sort of segregation that are different than the disomic segregation, the more diploid type segregation that is across the rest of the genome. So it's mostly simple, but also there are some regions that are complex. So it makes it really interesting. In terms of divergence of the A and the B genome or the subgenomes, that's happened relatively recently for many polyploid crops, about 2.5 million years ago. And so that's a lot younger and a lot more recent than wheat or brassica or other major polyploids. And the actual event where the 2 wild species came together and formed cultivated peanut is so young, it's actually very hard to age. It's somewhere less than 10,000 years ago that happened. And so cultivated peanut hasn't had a lot of time to accumulate new mutations to drive genetic diversity within the species. And so we call that this extreme genetic bottleneck, where a small population arose a new species, and it's so young that there hasn't been a lot of genetic diversity that has been driven in that species. And so pretty much peanuts that are grown in Australia, India, China, United States, Africa, and South America are extremely similar to one another, which makes crop improvement a little more challenging because we don't have a lot of extant diversity to draw upon. And so when we started talking about utilizing marker-assisted breeding in peanut, we really were focused on utilizing what we call QTL sequencing, which is this really great technique, which relies on bulk segregant analysis. And bulk segregant analysis is an old idea where if you have a structured population, in this case a simple biparental population, and these are images from a paper in 2013 on rice that was really the first paper to describe utilizing whole genome sequencing in plants for this analysis. And so if you have a population where the parents are different at a particular trait, for instance, this is just plant height, and you make a cross and have those individuals that are the progeny of those parents, there's a distribution of plant height within there. And so the hypothesis is if you take the extreme ends of those distributions, so the individuals that are the smallest and the individuals that are the tallest, and you do a bulk a DNA extraction and then sequence whole genome of that bulk DNA, you're actually profiling the allele frequency of those particular individuals, and you can look at them relative to one another. So the bulk of the tall individuals and the bulk of the short individuals, and if you look at them with each other, you identify a peak, which the hypothesis is that within this peak is enriched for alleles in one bulk and the opposite alleles in the other bulk. And so that is the genetic signal of this genetic region is actually controlling this trait that you're interested in. And why we were really excited to use this technology is that in peanut, because of sort of the complex nature and because it's a minor crop, for many years, breeders and geneticists didn't have access to markers to do the really high resolution genetics that other crops were doing. And so what they did was they focused on gathering really strong phenotypes on populations, in some cases across many environments and over many years. And so they had really strong phenotypic data sets. So we could go in and look at those data sets and make bulks based on them and do the sequencing quickly and identify regions to use in marker-assisted selection. And so this is an example of white mold or stem rot in peanut, which is a very important disease in the southeast United States, where there was a population where plant pathologists had phenotyped it for 6 years. And so we looked at the phenotypes and identified bulks and sequenced them. And we identified 2 QTL that we could validate as if you just select for these genetic regions in the population, you actually got the predicted phenotype. So a more resistant plant with one allele, a more susceptible plant with the other allele. And the picture at the bottom is one of my favorite pictures I like showing growers and stakeholder groups. These plots were manually inoculated with white mold, and we selected for them just with the markers and put them out inoculated with white mold. And you could see the one on the left that is essentially the susceptible line is completely dead. This is at the end of the year. And the other one that we selected to be resistant has peanuts on it and is very much thriving, even under manual inoculation of the pathogen. And so if we go back to the grass themselves, we see that they're very noisy. And actually, if you're a geneticist, you're thinking to yourself, I don't see a lot of resolution where I can pinpoint exactly where the genetic structure of these QTL. And that's because we had to utilize early informatics pipelines that took sequence of parental data first, called high-quality SNPs from those, and then profiled them in the bulk sequencing. So it added time, it added cost, and added an additional layer of complexity. And we still had sort of a lot of noise that we had to deal with. So I wasn't satisfied with that. We wanted to improve the informatics dramatically to the point that we didn't need to look at parental sequence. We could profile the genetic structure straight from the bulk sequence, and that would give us the ability to analyze data sets that from breeding programs where there was complex parentage, multi-parent structured data sets, as well as data sets where, quite frankly, sometimes the breeder has a hard time remembering who the parent actually was of that population, but there's a clear segregation of genetics. So that was one of the things that we did here when I started my group at Hudson Alpha 2 years ago, was we wanted to focus on improving those informatics to the point where we could do these analysis in a much better way. And so we did that, and we reanalyzed these data. We reanalyzed many data sets, but I'm just showing you the data that I showed as an example. These are the two QTL there. This is straight from the bulk sequence. There's much less noise, or much higher accuracy. So we no longer need parental sequence involved. And it gives us a lot more flexibility to analyze data sets, and that's really what we did. We went back and were able to analyze data sets from breeders that they couldn't do much with, and identify markers for them for the traits that they were interested in. So this was really pretty exciting development for us. The other part of this, that a lot of times when we look at QTL-seq data sets across different species, you're really just identifying the major loci, and so that's also not ideal. We want to be able to identify the minor loci as well, get the whole genetic structure for that trait. And so when we reanalyzed the stem rot data set, we identified a QTL that in our previous analysis we didn't find. And we were able to go back into the validation population, make selections for that particular QTL, and show quite clearly that there is a small effect of this loci, and actually is about 3%. And so that was really exciting, because now we have the confidence that the peaks that we see correspond to true genetic signal, instead of before sort of wondering, is this true or not? And it gives us the ability to identify these smaller effect QTL to make our breeding and improvement process that much more accurate and successful. So, peanut is what I mostly focus on, and it's my number one love in crops for many reasons. But we thought this was really successful in peanut. We should see how successful it would be in other complex crops. And so we looked at a data set that was published in 2019 looking at Brassica. And this was actually the group that published the very first QTL-seq paper. So they're really familiar with this analysis. This is a brassica that is heterozygous, so more like, it's not inbred like peanut or maize or wheat, but it's heterozygous, more like human beings or animals. And it also isn't self-fertile, so it outcrosses. So that means there's a lot more diversity, there's a lot more complex genetic structure happening. So you can see in column A, that's their original QTL-seq analysis. It's very noisy. They couldn't identify clear peaks. So what they did was they went back and they sequenced the parents of that population, and then they sequenced the F1 hybrid, and that represents filter 1 and filter 2. And that gave them the ability to identify the QTL for this particular trait. I think this is flowering time that they were interested in. And so that worked really well, and that's great, but that adds on cost and time, and in some cases you don't have the F1 hybrid to sequence, you don't have the parents to sequence, so that really makes it more difficult to be flexible. So we wanted to see, as our new informatics, could we go back and just look at those bulk sequences without all the other filters and identify those same QTL? And in fact, not only could we do that, but we did it in a more accurate way, less noise. And so that really showed me that what we had was something that could be broadly applicable across different crops and even animal species. So we're really excited about that. And so for someone like me, who's a genomics-focused crop improvement researcher who has spent time in industry and is really interested in real applicable crop improvement, we're always searching for the ability to genotype populations better, faster, and cheaper, right? Higher resolution, with more ease, and with less cost. So the best way to do that is to get very low coverage whole genome sequencing on your individuals. But the informatics involved with accurately making genotype calls at that level is very complex. So we felt like what we just developed was something that could be utilized in this way. So we went back to a population of 140 individuals. This is in peanut, that was segregating for tomato spotted wilt virus resistance, which is another virus that's very important in the southeast United States for peanut. In 2012, as part of a genome initiative that was supported by the industry, this population was sequenced to 5x coverage. At that point, with the cost of sequencing and the amount of sequence, because peanut genome is about 2.8 gigabases, about the size of the human genome, that was a pretty large cost to do one population at 5x coverage. I can't in the six-figure range. So that's very expensive. But we have the data. And initially, I used old pipelines, and we developed it, and were able to call SNPs, and we mapped them and identified a strong gene for TSWV resistance. It was very successful. And it represented a case study for us to say, can we use our new informatics to do this better at lower coverage? So we took that sequence and we downsampled it tenfold down to 0.5x coverage, meaning we have sequence covering for each individual about half the loci, just one read across the genome. Using the new informatics, we were not only able to analyze these data effectively, but we called about 9 times more SNPs and put on the map. And it was over 99% accurate. And if we look at the color map there, that's just showing the physical genotypes. Red is parent 1, blue is parent 2, and on one chromosome, where the individuals are actually in the columns and the markers are in rows. And so a geneticist that is used to looking at these types of things can see very clearly that there's no odd recombinations in there. It's very clean. We see that usual long stretch of recombination suppression in inbred crops. That's very common, not just in peanut, but in others. And so, the first time I saw this, I was hooked on, okay, we really have something here that we can utilize low coverage sequencing. So how do we do that, right? So a whole genome sequencing on a lot of individuals, the most expensive step is really doing the library prep. That's the most expensive part. And if you have to shear DNA and then do a regular Illumina library prep to get the sequencing, that would make it not actually cost effective at all. And so a colleague recommended using the Riptide library prep kit, and we ordered it, and after our first run, we never looked back. This library prep is very easy. It's easy to learn. It's easy to be highly reproducible. But what we really like about it is that there's only one step that's in the 96 well plate. So the way this works, really, it's a random primer-based library prep. You have your samples in a 96 well plate. Primers that are bar-coded are in those plates. Once the extension happens there, in the first step, you actually put all of the samples into one tube, and after that, everything's in one tube, which makes it very easy to scale up. At that point, there's a plate-based barcode on the other side that is attached to the Illumina barcode. There's an extension, an amplification, and then the size selection is just used with beads. And really, we're getting between 400 and 600 base pairs for our size selection every time. So it's really been really good, and we've utilized thousands of samples now. So really excited about this, and it's really opened up the door for us to do really neat things. And the first thing that we realized was that the whole thought about how you analyze QTL-seq data has now just been turned out on its head. Before, researchers, and I got a lot of questions about this because I was analyzing a lot of these data sets. Oh, how many individuals should I bulk? Which individuals should I bulk? Because you have one shot at it and you spend however much dollars on the sequencing. And if you get it back and it doesn't work, you have really no idea whether it didn't work because there's no genetic signal, your phenotypes are poor, you bulked the wrong individuals, you bulked the wrong amount of individuals. And so if you have a population that you phenotyped and you just do low coverage sequencing on the entire population and you have about 1x coverage, you already know that when you bulk 50 of those individuals or 20 of those individuals, each one is going to be represented about the same across. And so you have this sort of auto-normalization, and you can do multiple bulkings to look for the type of resolution that you're interested in. And so the actual informatics that we've developed to do all of that on the back end is very simple and easy and fast. And so what I'm showing here is just this region for the TSWV resistance from the peanut population we talked about before, the the signal is on the front end there on chromosome A1. You can see that little hump there in the 10%. On the bottom is a chromosome where there's no genetic signal, just as to show you the noise, so the noise on the other chromosomes and what that looks like. And so as you move forward into bulking more and more of the population, so even at 40%, so 40% susceptible, 40% resistant, so you're only not bulking 20% of those individuals. What happens is you have a lot less noise, you have tighter signal, but your genetic signal appears, it lowers because you're introducing more false negatives in there. So this gives you really a lot of flexibility, and it's not just one trait. If you have a population or a multi-parent population where you phenotyped dozens of traits that are strongly segregating in that population, you can easily map all of those traits within a few days once you get the sequence of all the individuals. So this is a completely new and powerful idea. And we've been using it, and we love the way that this works. So if we took it as a simple example, this is just one simple example that we've been working on this year. Just a simple biparental population with a resistant parent and susceptible parent. We identify that QTL using sequencing of the population. Because we have whole genome genotypes of every individual, we can go back in and search for informative recombinants, do a one step map and fine mapping all at one time. That points us to a region. What we usually do is get a hi-fi genome of our resistant parent or our parent with the phenotype that we're interested in. Usually in peanut, we see a large structural variant. We can validate that, and now we have functional variation and gene discovery in a more rapid sort of time than before if we were trying to layer SSR markers or layer GBS markers onto maps and map things. So this really speeds up the process and allows for the discovery of genes and functional variations that can be applicable in breeding programs. And that's really what our focus is. So this is pretty exciting. And then now we're pushing the envelope, okay? So a recombinant inbred population, that's good. But what if we just start looking at early generations and phenotyping one time? And so this is something that I'm exploring quite a bit. This was a collaborator that we looked at smut resistance, and smut resistance in peanut is really important because smut has the ability to move all around the world very quickly because the spores easily attach to equipment and they can survive harsh temperatures and pHs. And so they had a resistant parent, quickly made an F2 population. They phenotyped 600 F2s in the field, sent the DNA here, and we sequenced it and identified this really beautiful QTL on chromosome 12 here of peanut that we're now sort of validating and following up on our candidate gene. And so this makes the whole process even faster. So a lot of the projects that we're interested in, we're looking at early generation mapping, sequencing the largest number of individuals that we can, and really moving up the process quite a bit. And the other part that I just love is the ability to have this built-in error correction. So this is an example of a collaborator that we were looking at a trait where they first came to us and said, well, we have a qualitative score that we're interested in, and we did the bulking based on that. And you can see that maybe you can trick yourself into thinking there's a QTL there, there's genetic signal there, but it's not strong enough that I want to base a lot of follow-up work on. So I went back to them and they said, well, I also have this quantitative score. And when we bulk based on the quantitative score, now we get this beautiful signal that points to a very small region. And that really highlights this power that having the sequence of each individual gives you. It gives you the ability to really explore the data in a meaningful way, and you don't have to, I made a mistake on the front end and that's it, I shot it, and now I can't get it back. And so this was really an exciting thing to see, and highlights why what we're doing is so powerful. So we really wanted to take these gains and to move them through not just our collaborating partners, but give the ability for small groups to utilize genomics in this way across the world, basically. And so we developed a platform we call Kufu, which is essentially uses the Riptide library prep to get the front end sequencing and genotyping, and the informatics are on the back end that we can do all the other things for the genetics for the groups. So normally, if you are interested in doing whole genome sequencing, imputation, low pass sequencing, and you're part of a large commodity group that is going to run tens of thousands of samples, you can come together and go to companies that are extremely good at doing this, but they have to develop panels. There's upfront custom costs that have to be paid. And small groups that are working on crops that don't have the ability to do that, there's no way they have access to that. They have no access to that. But for us, we do completely panel-free imputation, low-pass whole genome sequencing and trait mapping, which I just described. And there's no panel that we need to construct. We don't have to have a custom design for capture or anything. We can utilize any crop that has a genome available. And so all we need to do is get the DNA, and we can do all the analysis for you. So it really has opened the door for groups that didn't really think that they could do this. Now they can do it. And I'm showing Waleed, who's my computational biologist, who developed all of the informatics on the back end of this, and then Courtney Valerio, who's very talented, who does all of the Riptide library prep. Thank you.